Morning everyone. Myself Dr. Vishwa Raj Lal, Faculty of Biotechnology, Jamshedpur Women's College, Jamshedpur. Today we will discuss about
Urine. In this, we will discuss about introductions, average composition of urine, GM per 24 hours, formations of urines, ultrafiltration, selective absorptions, secretions. In ultrafiltrations, we study filtrations fractions. In selective absorptions, Bowman's capsules, proximal convoluted tubule, loop of Henle, the thin walled portions, and collecting duct. And lastly is secretions. So, urine is a liquid byproduct of metabolisms in humans and in many other animals. Urine flows from the kidney through the uterus to the urinary bladder. Urination results in urine being excreted from the body through the urethra. Average amount of urine excreted in 24 hours 40 to 60 ounce. Urine examinations helps in the diagnosis of various renal as well as systematic disease. Normal urine is pale yellow due to presence of pigment urochrome. When the output of urine is low, it appears deep yellow. Normal odor of urine is aromatic in nature. Normal pH of urine ranges from 5.5 to 6.5. This average composition of urine in 24 hours. Water 1500 ml. Inorganic water 1500 ml is 95%. In inorganic substance, sodium 4, potassium 3, calcium 0.2, magnesium 0.1, iron 0.005, chloride 9.5, sulfate 2, phosphate 2, bicarbonate 1.5, ammonia 1, and in organic substances present urea 25, creatinine 1.5, uric acid 0.8. Next we come to our formations of urine. When urea enters glomerulus from liver through blood, the process of urine formations begins. The formation of urine is completed in following three steps. These steps are ultrafiltrations, selective absorptions, and secretions. Ultrafiltrations. We have studied that the kidney is composed of a large number of uriniferous tubules. Each uriniferous tubule is a delicate, very small coiled tube. Its terminal end has the Bowman's capsule, which surrounded the glomerulus, a bunch of blood vessels. The glomerulus is formed by afferent arteriole and efferent arterioles. The diameter of afferent arteriole is more than the diameter of efferent arteriole. So the blood pressure in the blood vessels of glomerulus is high. This pressure is termed hydrostatic pressure. In glomerulus, the hydrostatic pressure is about 75 mm Hg hemoglobin. In contrast to this, the pressure is only 30 mm Hg in Bowman's capsule. As a result of this much difference in hydrostatic pressure, about 1/5 of blood plasma, 20% comes to Bowman's capsule by filtrations. Along with blood plasma, all the substances which are dissolved in blood also reach the Bowman's capsule. This type of filtration which takes place under pressure is called ultrafiltrations. The effective filtration pressure that causes ultrafiltration is termed by three pressures. Filtration fraction. First, the fluid that filter out from blood into the Bowman's capsule of nephron is termed glomerular filter (GF). The glomerular filtrate formed in 1 minute in all the nephrons of both the kidneys is termed glomerular filtration rate. In a normal adult person, it is 125 ml of plasma fluid per minute. Third, on an average in a person having 70 kg weight, about 4.5 liter blood is present which is pumped through heart is 1 minute and received back. Out of this, only about 25% of it, that is 1250 ml, circulates through the kidney per minute. Out of this blood, only 650 ml is blood plasma, which is called renal plasma flow, RPF. Thus, we absorb that about one by fifth of the blood plasma filter out in kidney nephrons per minute. Thus, this rate of GFR and RPF is called filtration fractions. The fluid which reaches the Bowman's capsules after filtration is called glomerular filtrate or nephric filtrate. The glomerular filtrate contains the useful substance like glucose, salts, proteins, amino acids, etc. Only colloidal proteins cannot filter through the wall of blood vessels and wall of Bowman's capsule because their molecules are of very large. If all these substances go out of the body along with urine, the body will become weak and there will be hardly any use of ingesting food. Therefore, nature has a provision of selective reabsorptions of glomerular filtrate. Selective absorptions. The glomerular filtrate passes gradually from the proximal convoluted tubule, Henle's loop and distal convoluted tubule and reaches the collecting tubules. During the passage, the glomerular filtrate turns into urine by several chemical changes. When GF passes through the tubule, the 99% useful substances are absorbed by the blood vessels which surround Henle's loop. Here, urea and uric acids are not absorbed and thus this absorption is called selective reabsorptions. Some substances like water and glucose are absorbed in large quantity. Selective absorptions depends on the nature of substance and it's needed by the body. Selective absorptions of water takes place by simple process of osmosis. This does not need energy. Base dash water, some useful ions Na plus, Cl minus, SCO minus 3 are absorbed by active transport. So energy is required to absorb this ion. It is accomplished in following steps. First, Bowman's capsules. Second, proximal convoluted tubules, loop of Henle, the thin wall portions and collecting duct. To first, Bowman's capsule. Since the chemical compositions of glomerular filtrate is almost the same as of blood plasma, the glomerular filtrate is almost isotonic to the blood plasma. Proximal convoluted tubule. The proximal convoluted tubule is lined with columnar epithelial cells having microvilli and forming brush border and thus increasing the absorptive surface about 20 times. It is very suitable for absorptions and about 65% to 85% glomerular filtrate is reabsorbed into the blood present in peritubular capillaries through this epithelium. Glucose, amino acids, vitamins, ketone bodies, acetic acid, uric acid, chloride, sodium, potassium, phosphates, etc. are reabsorbed into the blood by simple diffusions and active transport. Water is also reabsorbed in blood by osmosis and thus osmotic pressure in the glomerular filtrate remains almost the same. That is isomotic to the blood plasma. Next, loop of Henle. It is distinguished into three parts: is the thin wall portion of the descending limb, the thin wall portion of ascending limb, and a thick wall part of ascending limb. The thin walled portion of depending limb is permeable to water and not to sodium and other solutes. Thus, in this part, more water is absorbed due to the increasing osmolality of intestinal fluid. In this way, the glomerular filtrate becomes concentrated and is therefore becomes about 4 times of hyperosmotic than blood plasma. Next, the thin wall portions of ascending limb of Henle's loop is poorly permeable in water, moderately permeable to sodium chloride, and is partially permeable to urea also. The filtrate therefore lose Na plus and by diffusion and gains in urea concentrations. Here it becomes isoosmotic again without any change in its volume. The wall of the distal convoluted tubule is very purely permeable to both urea and water and purely permeable to NaCl. In this region, the epithelial cells takes up chloride ions from the filtrate and expel this into interstitial fluid by active transport. Sodium ions also move out along with chloride ions. Thus, the filtrate become hypoosmotic. Next, collecting duct. From Henle's loop, the filtrate passes through collecting duct, which is all permeable to water. Its distal part, which is situated deep into the medullary pyramids, in permeable to urea. Thus, filtrate lose water and urea in this part and is finally converted into urine. The above discussed transport of substances facilitated by the special arrangement of Henle's loop and vice versa is called the counter current mechanisms. And lastly is secretions. The proximal convoluted tubule also alters the composition of glomerular filtrate by a process of secretions in its distal part. The excretory substances which are left in the uriniferous tubules are absorbed by the cells of these tubules, and these cells excrete additional waste from the blood stream into filtrate by active transport. This process adds urea, creatinine, hippuric acid, potassium and hydrogen ions, ammonia and little uric acid. Removal of hydrogen ions and ammonia from the blood helps to maintain a proper acid base balance in the body. Moreover, the drugs and pigments are excreted by tubule secretions. So this is about urine. Thank you.